Hello everybody. Now today we'll deal with the very important organelle mitochondria. Mitochondrion, also referred to as mitochondriae, mitochondriae in singular, in plural form, plural form we say mitochondria, in singular form we say mitochondrion. So this is the very structure of mitochondrion, and this is basically a sausage shaped cell organelle which is trapped inside the cell, which is actually was a bacteria or a bacterial prokaryote. Very first it got encapsulated within the eukaryotic cell, and now it exists as a cell organelle, but it is a different cell. It is referred to as a cell within the cell. See, I know that you wouldn't have understood. Let me tell you that this mitochondria existed as a single cell. Very early, I'm talking millions of years ago, a mitochondria was capable of existing alone as a cell. But then it got cocooned, it got surrounded by other cell, and it got trapped inside the cell. This prokaryote got trapped within the prokaryote or any eukaryote to form a cell organelle. So it is referred to as a cell within the cell, mitochondria. Mitochondria, as you know, is a double membranous structure. This is the outer membrane. This is the inner membrane. This is the inner membrane. Inner membrane bears infoldings. You can see that it is bearing certain folds. Inner membrane, like you see, I draw a simplified version. This is the outer membrane. This is the inner membrane. Fine. This space is the intermembrane space or perimitochondrial space, and here, like I draw the boundaries also, here exists the matrix of the mitochondria. So it's important. Like you see that here also fluid is present, aqueous fluid is present. Here two matrix is present. These foldings are called cristae or cristae, whatever. And these cristae are studded with specialized particles called oxysomes, or particles of Fernandez and Moron, or the particles of, or F1 F0 particles. So by different names they are known. They are studded on cristae. They are foldings like this. Basically, as you know, that it will have its own DNA also, because it was originally a cell, so it will have its own DNA, a double-stranded DNA. Double-stranded DNA. Also, it has its own 70S ribosomes. 70S means 50S and 30S. Don't get confused by this term. You'll get to know about this later on, but this is 50S, 30S, total 70S. I'm not doing addition 50S plus 70S. Please don't let that come into your mind for a second also. This is a stranded DNA, a double-stranded circular DNA, which is encapsulated over here. Cristae are their outer inner membrane. This intermembrane space is there, inner membrane is there. Inner membrane has enzymes like succinate dehydrogenase, and basically mitochondria has very important role to play in the cell. I'll tell you how does it play. Let me tell you. Wait a minute, just wait a minute, please. Give you the brief. Wait a second. Let you know what it is all about. See, this is a section view that I was showing to you. Let me take my these also. See this or this, whichever you prefer. Fine. Crystal matrix, inner membrane, outer membrane. These particles of Fernandez-Moran, F1 particles, oxysomes, cristae, like it is all about that. So you can see the magnified version also. Intracrystal space. Yes, this is left. That this space is referred to as the intracrystal space. F1, F0 particles, cristae, these foldings cristae, and here enzymes are there for the protein synthesis, lipid synthesis, and the Krebs cycle also. We know single circular, double stranded molecule, GC content, which I was talking of, this DNA which is present. Also, we came to know that it is gonna have 70s ribosomes and a few RNA molecules also. These are RNA molecules. Fine. See, this is this oxysome or F0 F1 particle. It has enzymes. Mitochondria, as you know, site of aerobic respiration. You would have done in 10th class also. And they are the powerhouse of cell because ATP synthesis is over here. It helps in oxidative phosphorylation coupled with the release of ATP. You'll read about it later on in photosynthesis chapter. ATP synthetase enzyme is there. Many different enzymes, matrix, TCA enzymes, carboxylic cycle, different things are there, which is actually all about theory. You can read it from the NCERT to know better. Thank you.